1. Synthesizing life, the ambitious project to create a human chromosome from scratch. 2. Moonshot science, building a synthetic human genome from scratch. 3. Artificial human genomes, first major step achieved synthetic DNA breakthrough. 4. £10 million project to build synthetic human genome, the future of genomic science. 5. Synthetic human genome project, rewriting DNA to revolutionize medicine and biology. 1. Synthesizing life, the ambitious project to create a human chromosome from scratch. Imagine a world where scientists don't just read the blueprint of life, they rewrite it. That's exactly what a groundbreaking new project is attempting to do by synthesizing an entire human chromosome from scratch. But here's where it gets controversial. While we've made incredible strides in genetic research, the idea of creating life's building blocks in a lab raises profound ethical and scientific questions. Are we playing God, or simply unlocking the next frontier of medicine? Let's dive in and explore the fascinating, and sometimes unsettling, details of this ambitious endeavor. When the first draft of the human genome was revealed in 2000, it was hailed as a monumental achievement. Then President Bill Clinton famously declared that humanity was learning the language with which God created life. Yet, as it turns out, that declaration was a bit premature. The complete sequence wasn't finalized until 2022, and while we've mastered reading DNA and even making small edits with tools like CRISPR, writing the genomic code from scratch has remained a daunting challenge. Think of it like this. Reading a book is one thing, but writing an entirely new chapter in the same language is something else entirely. The project to synthesize a human chromosome represents a bold leap forward in genetic engineering. Scientists have long been able to sequence DNA, which is akin to reading a complex instruction manual. They've also used CRISPR to tweak specific genes, much like editing a few lines of code in a computer program. However, constructing an entire chromosome, a structure containing millions of genetic letters, is akin to writing a whole new book from scratch. This isn't just about making minor adjustments. It's about creating something entirely new, with implications that could revolutionize medicine, agriculture, and even our understanding of life itself. But this is the part most people miss. The ethical dilemmas are just as complex as the science. If we can synthesize chromosomes, where do we draw the line? Could this technology be used to create designer humans, or even to resurrect extinct species? The possibilities are as thrilling as they are unsettling. And while the scientific community is buzzing with excitement, the broader public is left grappling with questions that don't have easy answers. What do you think? Is this a step toward curing genetic diseases and extending human life? Or are we venturing into territory that should remain untouched? The debate is just beginning, and your voice matters. Share your thoughts in the comments. Are we on the brink of a medical revolution? Or are we tampering with forces we don't fully understand? 2. Moonshot science, building a synthetic human genome from scratch. Imagine building a human genome from the ground up, like assembling a complex, microscopic Lego set. It sounds like science fiction, but it's the ambitious goal of a groundbreaking new initiative. We've already witnessed the incredible impact of the Human Genome Project, which mapped our entire DNA sequence. This monumental effort, spanning over a decade and involving thousands of researchers worldwide, has revolutionized our understanding of disease and fueled advancements in DNA sequencing technology. The cost of sequencing a human genome has plummeted, making it more accessible than ever. But here's where it gets even more exciting. What if we could not just read our DNA, but build it? The Wellcome Trust, a global health research foundation, is investing £10 million, approximately $13.6 million, to launch the Synthetic Human Genome Project, SynHG. This audacious project aims to synthesize entire human genomes from scratch, potentially unlocking even more profound breakthroughs in biomedicine and biotechnology. Leading this ambitious endeavor is Jason Chin of the University of Oxford, whose team previously achieved the remarkable feat of synthesizing the entire genome of the E. coli bacteria. This, coupled with the recent successful synthesis of a yeast genome, a significantly more complex organism closer to humans in evolutionary terms, provides a strong foundation for the SynHG project. And this is the part most people miss. While gene editing allows us to modify existing genetic code, synthesizing DNA allows us to build entirely new sequences. Think of it like renovating a house versus building one from the ground up. This opens up a world of possibilities, from deeper understanding of diseases to the creation of designer cells, tissues, and even organs. Matthew Hurrells, director of the Wellcome Sanger Institute, explains that building DNA from scratch allows us to test theories about how DNA functions in ways that simply tweaking existing DNA can't. Currently, our understanding of the human genome is largely limited to the 2% that codes for proteins. The remaining 98%, often referred to as non-coding DNA, remains largely mysterious. SynHG aims to shed light on this genomic dark matter, potentially revealing crucial insights into its function and influence. But here's where it gets controversial. The ability to synthesize human genomes raises significant ethical concerns. Could this technology be misused to create bioweapons or genetically enhanced humans? Some even fear the creation of novel organisms incorporating human DNA. Geneticist Bill Earnshaw aptly points out, the genie is out of the bottle. If an organization with the right resources decides to synthesize something, I don't think we could stop them. This raises a critical question. How do we balance the potential benefits of this technology with the risks? Recognizing these ethical dilemmas, SynHG includes a social science program to explore potential risks and develop strategies to mitigate them. One key focus is the current bias in genomic research towards individuals of European ancestry, which could limit the broader applicability of findings. Do you think this focus on ethical considerations is enough? What other safeguards should be put in place? Share your thoughts in the comments below. Despite the immense technical challenges and ethical considerations, the potential rewards of SynHG are enormous. With a timeline of 5 to 10 years to synthesize a single human chromosome, and likely decades to complete an entire genome, there's time to carefully navigate the ethical landscape. If successful, this project could ignite a second revolution in genetics, promising advancements that could dramatically improve human health and well-being. What are your hopes and concerns about this groundbreaking endeavor? Let us know in the comments. 3. Artificial human genomes. First major step achieved synthetic DNA breakthrough. Imagine rebuilding the entire instruction manual for human life, letter by letter. That is exactly what an international team of researchers has just set in motion, and the implications reach far beyond the walls of any single laboratory. The new initiative, called the Synthetic Human Genome Project, SynHG, dares to move beyond merely reading our 3 billion letter code. Instead, it plans to write a piece of that code completely from scratch. Phase 1 aims to design, purely in silico, and then chemically assemble one whole human chromosome, a DNA macromolecule that represents roughly 2% of everything that makes us biologically human. To achieve this, computers will first draft an uninterrupted digital sequence. Advanced robotic systems will then stitch together billions of chemical building blocks to create a single, ultra-long strand of synthetic DNA. Scientists and bioengineers at Oxford, Kent, Manchester, Cambridge, and Imperial College London believe this proof of concept could be ready within the next 5 to 10 years. With a fresh £10 million, about $13.7 million, infusion from the Wellcome Trust, their optimism feels palpable. Some would even say audacious. And this is the part most people miss. If the plan succeeds, it could unlock a toolkit for custom-built tissues that resist viruses, personalized cell therapies that outsmart cancer, or even enzyme tweaks that fight aging. Project leader Jason Chin argues that the moment we can build a chromosome on demand, we will finally be able to test, rather than merely theorize, how specific stretches of DNA influence health, disease, and evolution. But here's where it gets controversial. Not everyone is convinced the timeline is realistic. Robin Lovellbadge, a decorated geneticist at the Francis Crick Institute who is not involved in SynHG, applauds the ambition. You only truly understand something if you can build it, yet remains skeptical. After all, the most complete lab-made genomes to date belong to single-celled organisms with just 12 million base pairs spread across 16 chromosomes. Humanity's blueprint is 250 times larger and divided into 46 chromosomes that must function in more than 30 trillion human cells. Scaling up is a mountain no one has climbed before. Some critics point out that even if scientists could assemble an entire set of human chromosomes, we are light years away from constructing a safe, fully synthetic human being. Revolutionize bluntly states we have no idea how to do that, and if we tried, it is likely to be very unsafe. To preempt ethical backlash, the SynHG Consortium says it is already consulting policy experts, industry leaders, civil society groups, and ethicists. They recognize that questions about eugenics, reproductive rights, and so-called designer babies will resurface. Sarah Norcross, director of the Progress Educational Trust, stresses the need for open dialogue so that the public grasps what scientists are and are not proposing. Yet skeptics worry that a handful of prestige labs could steer the conversation before wider society catches up. If synthetic genomes can be patented, will corporations dictate which gene edits become available or affordable? Might governments impose restrictions that vary dramatically between countries, fostering a global genetic divide? Bold claim or empty promise? Supporters counter that past Moonshot projects, the Human Genome Project itself or CRISPR genome editing, also faced intense doubt before becoming standard scientific tools. They suggest SynHG could similarly transition from fantasy to routine practice, accelerating biomedical breakthroughs in cancer immunotherapy, viral-proof organ transplants, and perhaps even reversing certain genetic disorders before birth. And this is the part that could split the room. Is it ethically acceptable to redesign human DNA if the ultimate goal is curing disease? Or does any rewrite, however noble, open a slippery slope toward traits-based selection? Should society draw a clear line now, or wait to see what is technically feasible first? Your turn. Do you see SynHG as the next logical leap in biomedical science, or as a venture that risks outpacing ethics? Would you volunteer your cells for testing if it accelerated treatments? Where should we set the guardrails, on medical grounds only, or should personal enhancement ever be on the table? Jump into the debate below and let us know whether you're cheering, cautious, or downright concerned. For £10 million project to build synthetic human genome, the future of genomic science. Imagine a future where scientists can craft an entirely new human genome from scratch. A groundbreaking endeavor that could revolutionize our understanding of biology and medicine. But here's where it gets controversial. A £10 million project led by the prestigious Wellcome Trust aims to do just that, embarking on a 5-year journey to synthesize a human genome. This ambitious initiative, known as Synthetic Human Genome, SynHG, is poised to push the boundaries of what's possible in genetic engineering. At the turn of the 21st century, the Human Genome Project marked a monumental achievement by providing the first draft of the human genome in 2003. As Professor Richard Gibbs, a renowned human geneticist, put it, this project changed the way people thought biology could be done. Since then, the field of genomics has evolved dramatically, influencing both research and society. Parallel to reading genomes, scientists have been exploring the feasibility of writing them, essentially constructing a genome from the ground up. The Wellcome Trust's SynHG project continues this exploration, aiming to develop the tools, technologies, and methods necessary for human genome synthesis. According to Dr. Michael Dunn, Director of Discovery Research at Wellcome, this endeavor holds the potential to uncover new insights into health and disease, fundamentally altering our comprehension of life and well-being. But why create synthetic genomes? Physicist Richard Feynman once wrote on his blackboard, "What I cannot create, I do not understand." Although modern technology allows us to sequence an organism's genome in mere hours, many aspects of its function remain mysterious. Professor Robin Lovelbadge, a principal group leader at the Francis Crick Institute, highlights several gaps in our current understanding. While the protein-coding regions of the genome are relatively straightforward, they represent only a small portion of the total. The repetitive DNA at the ends of chromosomes, telomeres, and centromeres, crucial for chromosome segregation during cell division, remain less understood. Additionally, there are vast numbers of repetitive elements, some of which are remnants of ancient viral integrations or mobile genetic elements. Lovelbadge explains, these repetitive elements and their associated proteins play a vital role in the dynamic folding and organization of the genome, influencing not only chromosome packaging but also gene expression. Studying their functions is challenging and can sometimes reveal elements that are redundant or evolutionary leftovers, making the process time-consuming and potentially unfruitful. Addressing these knowledge gaps motivates the pursuit of synthetic genomes. Unlike genome editing tools such as CRISPR-Cas9, zinc finger nucleases, ZFN, and transcription activator-like effector nucleases, TALENs, which allow for targeted modifications, synthetic genomes enable large-scale changes, offering a more comprehensive approach to genetic manipulation. Synthetic genomics, a branch of synthetic biology, focuses on creating viruses, bacteria, and eukaryotic cells with synthetic genomes. This involves designing and assembling DNA sequences that are then introduced into host cells. Early successes in synthetic genomics include the creation of the first synthetic bacterial genome, Mycoplasma mycoides JCVI Syn 1.0, in 2010, and a 4 megabase synthetic E. coli genome in 2019. In 2023, researchers from the Synthetic Yeast Genome Project 2.0, SC 2.0, consortium achieved another milestone by engineering a yeast strain with 50% synthetic DNA. This accomplishment is significant because yeast genomes are more complex and repetitive than bacterial genomes, consisting of multiple chromosomes rather than a single one. Given the close relationship between humans and yeast, synthesizing yeast genomes could provide valuable insights into human biology and disease. However, the ultimate goal remains the construction of larger, more intricate genomes, such as those found in crops or humans. As Lovelbadge notes, the ability to build and redesign segments or entire human chromosomes is crucial for true understanding and potential improvements in gene activity. Despite the potential benefits, the technology required to synthesize a human genome does not yet exist. The SynHG project, led by Professor Jason Chin from the Generative Biology Institute at the Ellison Institute of Technology and the University of Oxford, aims to develop the necessary tools over the next 5 to 10 years. Their initial focus will be on creating a fully synthetic human chromosome, with the eventual goal of developing methods to modify chromosome sequences with minimal impact on protein production. According to Lovelbadge, while the SynHG project may not reach the stage of creating synthetic human chromosomes in the near future, it could pave the way for lab-grown synthetic cells with high efficiency. Any use of these cells in humans would require careful design to prevent tumor formation or the creation of novel infectious agents. The SynHG project also acknowledges the complex societal and ethical implications of synthetic genomes. Introducing synthetic DNA into living cells could lead to unforeseen biological consequences, potentially disrupting ecosystems. Furthermore, the ability to recreate an individual's gene or genome sequence in a laboratory raises questions about genetic privacy and safety. To address these concerns, the SynHG team has launched a social science program called Careful Synthesis, led by Professor Joy Zhang from the Center for Global Science and Epistemic Justice at the University of Kent. This program will investigate the potential impacts of synthetic DNA creation, collaborating with academics, industry, civil society, and policy partners across Europe, Asia-Pacific, Africa, and the Americas. Careful Synthesis aims to establish a new paradigm for accountable scientific and innovative practices in the global age, combining technical possibilities with diverse socio-ethical perspectives, Zhang states. Sarah Norcross, director of the Progress Educational Trust, PET, emphasizes the importance of balancing ambition with diligence in genomic research. She warns that this work should not overshadow ongoing advancements in genome sequencing and editing, noting that the Human Genome Project was only fully completed in 2023, 2 decades after its initial draft. Similarly, the potential applications and consequences of genome editing are still largely unexplored. In conclusion, the SynHG project represents a significant step forward in the field of synthetic genomics, with the potential to unlock new knowledge and technologies. However, it also raises important questions about the ethical and societal implications of such endeavors. As we continue to push the boundaries of genetic engineering, it is crucial to proceed with caution and consideration. What do you think about the prospects and challenges of creating synthetic human genomes? Share your thoughts in the comments below. 5. Synthetic Human Genome Project, Rewriting DNA to Revolutionize Medicine and Biology. Imagine a world where scientists can recreate the human genetic code from scratch in a laboratory. It sounds like something out of science fiction, but it's closer to reality than you might think. This bold endeavor, known as the Synthetic Human Genome Project, SynHG, could revolutionize our understanding of biology and disease while paving the way for groundbreaking medical innovations, though not without sparking heated debates along the way. It has been just over 2 decades since the completion of the original Human Genome Project, a monumental achievement that decoded nearly all of our DNA and opened doors to countless advancements in medicine and gene editing technologies. At the time, simply reading the genome was an enormous challenge, costing billions of dollars and taking more than a decade to finish. Now, researchers are setting their sights even higher. Instead of merely reading or editing genes, they aim to write them entirely from scratch. But here's where it gets controversial and exciting. Recent breakthroughs in synthesizing the genomes of simpler organisms, such as yeast and bacteria like Escherichia coli, have convinced leading institutions, including the global health charity Wellcome, that humanity is ready to tackle the far greater challenge of creating synthetic versions of human chromosomes. While this project will take decades and require funding ranging from millions to hundreds of millions of pounds, its potential impact is staggering. To kick things off, Wellcome has already committed £10 million to support research teams at prestigious universities across the UK, including Cambridge, Oxford, Manchester, Kent, and Imperial College London. Over the next 5 years, these scientists hope to recreate one small chromosome, a mere 2% of the entire human genome, as a proof-of-concept step toward larger goals. The specific chromosome hasn't been chosen yet. First, they need to refine the techniques required to write and assemble long strands of DNA accurately. So why go through the trouble of recreating the human genome from scratch? For starters, the journey itself holds immense value. Along the path to synthesizing the full genome, researchers expect to develop cutting-edge tools and methods that could transform fields beyond genetics. For instance, being able to insert large sections of synthetic DNA into human cells could accelerate drug discovery and enable advanced cell therapies. Additionally, this work may shed light on how mutations in entire genes or clusters of genes contribute to complex diseases, offering clues to prevention and treatment strategies. And this is the part most people miss. The benefits extend far beyond human biology. The same tools used to synthesize human DNA could also be applied to other complex genomes, such as those of plants. Imagine engineering crops capable of thriving in extreme climates, ensuring food security for future generations. Such possibilities underscore the broader significance of mastering genome synthesis. However, the road ahead is fraught with challenges. To create even a single small chromosome, containing about 50 million base pairs out of the 3 billion in the human genome, scientists must overcome significant hurdles. One major obstacle is cost. Unlike DNA sequencing, which has become exponentially cheaper over the years, synthesizing DNA remains prohibitively expensive. Producing long strands involves screening for errors, isolating correctly synthesized sequences, and using vast amounts of organic solvents, much of which generates chemical waste. Some companies are working hard to address these issues by developing greener alternatives and improving efficiency. Another difficulty lies in dealing with non-coding DNA, often referred to as junk DNA. These repetitive sequences play roles in regulating gene activity or maintaining structural integrity, but they're notoriously tricky to synthesize and assemble. Current chemical methods allow scientists to produce stretches of a few hundred base pairs reliably, but longer sequences increase the likelihood of errors that must be painstakingly screened out. Some innovative approaches involve designing enzymes to assemble DNA segments up to several thousand bases, while others rely on cloning in bacteria, but these processes introduce errors too. Enter chemistry, the unsung hero of this ambitious project. Researchers hope that chemists can devise novel ways to produce ultra-long DNA strands, 20,000 base pairs or more, so they can focus on perfecting assembly techniques. Several commercial providers are racing to develop new sequence synthesis technologies that could not only facilitate the SynHG but also drive down costs significantly. Expanding applications for synthetic nucleotides are expected to further fuel technological progress. Of course, no discussion of synthetic genomics would be complete without addressing the elephant in the room: ethics. Right now, any synthetic DNA created under the SynHG initiative is strictly intended for use in lab-grown human cells, derived from a specific type of retinal pigment epithelium. But ethical concerns loom large, particularly regarding the potential misuse of this powerful technology. Could it lead to the creation of so-called designer babies? How do we ensure responsible use? To stay ahead of these questions, the scientific community is proactively engaging social scientists to explore the ethical, legal, and societal implications of synthesizing genomes. By fostering open dialogue about policy frameworks and safeguards, they hope to prevent misuse while encouraging innovation. Yet, opinions remain divided. Should we embrace the limitless possibilities of synthetic biology, or proceed with caution due to unknown risks? What do you think? Is the promise of curing diseases and securing food supplies worth the potential dangers? Or should we tread carefully, prioritizing regulation over rapid advancement? Share your thoughts in the comments below. We'd love to hear your perspective.